Welcome to another episode from the Veterinary Medicine Digest. Today, we delve into the intricate world of hearing loss in our beloved companion animals, exploring the surprising prevalence of deafness in both dogs and cats, the underlying genetic causes, and the fascinating ways their brains adapt when the sense of sound is diminished or lost entirely. Auditory function is essential for animals, influencing their interactions with the environment, their communication with human families and conspecifics, and their ability to detect dangers like motor vehicles or predators. A reduction or total loss of hearing, referred to as deafness, can range from a mild inconvenience to an extreme handicap, depending on the severity and whether one or both ears are affected. In veterinary medicine, recognizing and diagnosing deafness is crucial, especially since behavioral testing alone is notoriously unreliable. A dog may fail to respond to a sound due to situational anxiety, or conversely, a deaf dog might respond by detecting visual cues, vibration, or even air movement associated with the stimulus. Definitive diagnosis requires objective electrodiagnostic testing, specifically the Brainstem auditory evoked response testing, which is often referred to as a Brainstem auditory evoked potential. This specialized procedure measures the brainwaves produced along the hearing pathway after a sound stimulus, allowing clinicians to objectively determine if hearing loss is present and, if so, localize where the failure of electrical impulse generation occurs. The blueprint of hearing and the failure of transduction. To understand deafness, we must first appreciate the delicate complexity of normal auditory function. Sound waves, oscillating changes in air pressure, are collected by the external ear, channeled through the ear canal to the eardrum, or tympanic membrane, which begins vibrating. These mechanical vibrations are transferred through the middle ear bones, the malleus, incus, and stapes, to the fluid-filled internal ear. The internal ear contains the cochlea, a spiraled bony tube with three fluid-filled chambers, the scala vestibuli, scala tympani, and scala media. Movement of the stapes against the oval window imparts vibrations to the fluid, or perilymph, in the outer chambers, causing movement in the basilar membrane that borders the middle chamber, the scala media. Resting on the basilar membrane is the organ of Corti, the sensory organ for hearing, which contains specialized hair cells that are sensitive to bending movements. When the basilar membrane moves, the hair cells shear against the tectorial membrane, generating an electrical signal that is transmitted through the vestibulocochlear nerve, cranial nerve 8, to the brain. The proper function of the internal ear depends heavily on a highly specialized chemical environment maintained within the scala media. The fluid here, called endolymph, is rich in potassium and maintains a massive electrical gradient, known as the endocochlear potential, which can be around 180 millivolts. This large potential difference serves as the engine driving the sensory hair cell receptors. Crucially, the stria vascularis, a multilayered epithelial structure on the outer wall of the scala media, is responsible for maintaining these high potassium levels. This structure relies on the presence and proper function of melanocytes, the same pigment cells found in the skin and hair. Damage to the stria vascularis, or the absence of functioning melanocytes within it, results in the loss of this critical electrical potential, leading to secondary degeneration of the hair cells and, ultimately, sensorineural deafness. This is the central mechanism underlying pigment-associated hereditary deafness. Classifying deafness in animals. Deafness can be categorized based on its location in the auditory system, its origin, and its onset. Conductive deafness results when sound waves are reduced or blocked from reaching the internal ear, typically due to problems in the outer or middle ear. Common acquired causes include chronic inflammation or infection, otitis externa and media, excessive earwax impaction, cerumen impaction, foreign bodies, or middle ear polyps. Conductive deafness is often treatable and may be resolved with appropriate medical or surgical intervention, depending on the underlying pathology. A suspected hereditary form of later-onset conductive deafness is primary secretory otitis media, or glue ear, which primarily affects Cavalier King Charles Spaniels. Sensorineural deafness involves damage to the delicate hair cells or the afferent neurons of the cochlea. At present, sensorineural deafness in mammals cannot be corrected, though ongoing research hopes to change this through hair cell regeneration. This type of hearing loss can be congenital, present at or near birth, or later onset acquired. Acquired later onset sensorineural deafness can result from several factors. 1. Presbycusis, or age-related hearing loss. This is a progressive, typically bilateral form of sensorineural deafness that may include conductive elements and affects high frequencies first. It is commonly observed in dogs starting around 8 to 10 years of age in middle-sized breeds, leading to degeneration of the stria vascularis, hair cells, and spiral ganglion cells. 2. Ototoxicity. Exposure to certain drugs or chemicals is toxic to the internal ear, causing partial or total functional loss that is often permanent by the time it is detected. One of the most common culprits is the aminoglycoside antibiotic gentamicin, often used topically for otitis externa. Other classes include loop diuretics and antineoplastic agents. 3. Noise-induced hearing loss. This cumulative process affects military dogs, hunting companions exposed to gunfire, and pets housed in kennels with high ambient noise levels. Hearing loss results from the mechanical disruption and subsequent loss of hair cells. Because the damage is gradual and cumulative, owners often report an apparent acute onset when the animal can no longer compensate for the progressive loss. 4. Otitis interna. Infections that reach the internal ear can produce sensorineural deafness, often accompanied by vestibular signs. Prompt antimicrobial therapy is essential to minimize potential permanent damage. 5. Anesthesia-associated deafness. Although uncommon, some dogs and cats experience bilateral, permanent deafness following anesthesia, particularly after dental procedures or ear cleanings, though the specific mechanism remains largely unknown. In addition to direct hearing loss, animals may experience related auditory conditions. Tinnitus is the perception of phantom sounds, such as ringing or buzzing, when no external sound is present. While subjective tinnitus cannot be confirmed in animals, dogs with acute deafness sometimes show anxious behavior that might suggest its presence. Hyperacusis is an increased sensitivity to sounds that would otherwise not be bothersome, but studies on this condition in dogs and cats are lacking. Hereditary deafness in canines, the Dalmatian enigma. The most frequent clinical presentation of sensorineural deafness in dogs is congenital hereditary deafness. This condition is strongly linked to white or dilute pigmentation patterns and is generally classified as cochleosaccular type degeneration. Dog breeds with reported congenital deafness number more than 90, including the Australian Shepherd, Border Collie, Bull Terrier, Boxer, Dalmatian, English Setter, and Siberian Husky. This type of deafness is specifically associated with either the recessive alleles of the piebald gene or the dominant allele of the merle gene, both of which suppress melanocyte function. The piebald gene is encoded by the microphthalmia-associated transcription factor gene, MITF, located on canine chromosome 20. The merle gene is encoded by a mutation in the S-Helvy pigmentation gene on canine chromosome 10. These genes suppress melanocytes, leading not only to white or diluted pigmentation but also to disrupted function in the stria vascularis of the internal ear, which results in deafness. The risk of deafness in these breeds is often significantly associated with the absence of iris pigment, blue eyes. The Dalmatian breed presents the highest prevalence of congenital sensorineural deafness, affecting up to 30% globally. In Dalmatians, the condition is strongly associated with extreme white coat patterning, which is a breed hallmark trait linked to the microphthalmia-associated transcription factor gene. Dalmatians showing pigmented patches on their body or head, or those without blue eyes, appear to have a reduced deafness risk. Researchers have struggled for years to pinpoint the precise genetic basis of deafness in Dalmatians, despite numerous attempts. However, a recent and comprehensive genome-wide meta-analysis pooling data from Australian, United Kingdom, and United States Dalmatian populations, achieved a significant breakthrough. This extensive analysis confirmed a major association signal for congenital sensorineural deafness on canine chromosome 20, specifically in the vicinity of the microphthalmia-associated transcription factor gene. This crucial research identified a single, well-defined genome-wide significant risk haplotype for the disease. The risk haplotype was formed by 3 specific markers on canine chromosome 20. When examining the relationship between this haplotype and deafness, the data showed an increased bilateral deafness risk with allele dosage in an additive manner. Dogs who were homozygous for the high-risk haplotype were approximately 10 times more likely to be bilaterally deaf compared to dogs homozygous for the low-risk haplotype. Specifically, in the combined dataset, the frequency of bilateral deafness was 38% in animals homozygous for the risk haplotype compared to 11.7% in animals homozygous for the low-risk haplotype. The effect on unilateral deafness was less dramatic. Despite this strong association, the penetrance of the risk haplotype is considered modest, as only 38% of dogs homozygous for the risk haplotype were clinically deaf in the combined data resource. This fact, coupled with the finding that some dogs homozygous for the protective low-risk haplotype were also deaf, suggests that other genetic loci or additional variants at this location likely contribute to deafness in this breed. This research paves the way for potential genetic testing, which, in combination with existing selection criteria for breeding animals, could assist in reducing the number of deaf Dalmatians by selecting animals with the protective haplotype. Beyond pigment-associated deafness, other types of hereditary congenital sensorineural deafness exist. These are often categorized as neuroepithelial-type abnormalities, where deafness occurs due to hair cell degeneration after the normal pattern of development. Similarly, a specific line of pointers bred for enhanced anxious behavior concurrently developed deafness, also of the neuroepithelial type, suggesting an autosomal recessive inheritance pattern. The mechanisms and responsible genes for these non-pigment-associated neuroepithelial forms are generally still unknown. The feline model of deafness. Moving to our feline friends, cats are recognized for having some of the best hearing among mammals, being extremely sensitive to a broad range of frequencies. However, they are also famously affected by congenital deafness, particularly those with a white coat and blue eyes. Deafness in white cats is strongly associated with a dominant white locus, or W locus, which demonstrates pleiotropic effects, meaning a single gene causes multiple observable characteristics. These effects typically include the white coat, blue irises, and deafness. As with dogs, the underlying pathology is usually the absence or abnormality of melanocytes in the stria vascularis of the inner ear, leading to the degeneration of the cochlea and saccule, a condition termed cochleosaccular degeneration, which mirrors the shibe deformity observed in humans. It is important to note that the correlation between these traits is imperfect, a concept known as reduced penetrance. Not all white cats are deaf or blue-eyed. However, the likelihood of a white cat being deaf has been calculated at 80% if blue irises are present. The complexity is further shown by the fact that albino cats, who lack the enzyme tyrosinase required to produce melanin, have a normal distribution of melanocytes and are not deaf, distinguishing them anatomically from the typically deaf white cat. New insights into feline dominant blue eyes and deafness. Recent research has focused on genetic variants responsible for dominant blue eyes, or DBE, in various cat lineages, including the Maine Coon breed. This trait is associated with minimal white spotting and has been confirmed to be autosomal dominant, though different founding genetic effects have been identified. Specifically, researchers have identified multiple variants in the paired box3 gene, or PX3, which account for some of these feline phenotypes. The PX3 gene is a critical candidate because its human counterpart is involved in Waardenburg syndrome, which links pigmentation abnormalities with deafness. A newly characterized dominant blue eyes lineage in the Maine Coon breed, called the Agostino line, was found to carry a fourth paired box3 variant, a frameshift variant termed PX3C.160 deletion. This deletion is predicted to produce a truncated protein lacking a significant portion of the wild-type protein. All 13 dominant blue eyes cats from this line studied were heterozygous for the variant, confirming its dominant inheritance pattern, and it was perfect in genotype-phenotype correlation for the blue eye trait. However, breeders reported no deafness in this specific Agostino line, though formal brainstem auditory evoked response testing had not been performed. In contrast, an established Maine Coon line, the Rosarii elvis line, which carries the PX3C.937 cytosine to thymine variant, had previously been associated with deafness. Earlier data on 8 tested cats from this line showed all were deaf. However, a subsequent follow-up study conducting brainstem auditory evoked response testing on 10 additional cats from the Rosarii elvis line, 6 with dominant blue eyes and 4 without, revealed that none exhibited deafness. All 10 were bilaterally hearing. This result dramatically contrasts the earlier findings and highlights the potential influence of modifying alleles from the genetic background or other complex factors in the expression of deafness, similar to situations described in white-spotted dogs. The PX3 variant found in the celestial line was not associated with deafness in heterozygous cats, though deafness was linked to combined variants or homozygosity in other lines. Furthermore, the study improved the phenotypic description by revealing the existence of latent cats in the Rosarii elvis line. These latent cats are proven carriers of the dominant blue eyes variant through genotyping, but do not express the blue eye phenotype. In summary, regarding deafness associated with dominant blue eyes in cats, it is clearly linked to the Rosarii elvis variant, but the wide discrepancy in reported prevalence underscores the urgent need for further data to determine how often this deafness truly occurs. The central nervous system consequences of deafness. The loss of hearing does not just affect the cochlea, it produces widespread and profound changes in the central auditory system. Afferent input from the cochlear nerve is vital for the normal development and maintenance of brain structures responsible for sound processing. Sensorineural deafness leads to the degeneration of the spiral ganglion cells, which are the neurons that collect auditory information from the hair cells and relay it to the brain. In congenitally deaf white cats, there is a gradual loss of these ganglion cells over time. Beyond the peripheral structures, congenital deafness triggers changes in the brainstem, which is the gateway to the central auditory system. Structures like the cochlear nucleus and the superior olivary complex, which are critical for processing acoustic cues like interaural timing differences, sound location, show significant disruptions. Synapses in these areas, like the prominent end-bulb terminal in the cochlear nucleus, show distinct morphological changes, including proliferation of vesicles and thickening of postsynaptic densities, suggesting an adaptation or struggle to compensate for the missing signal. In the medial superior olive, a convergence point for input from both ears, congenital deafness causes a bilateral disruption in input, characterized by significant reduction in inhibitory input and shrinkage of excitatory inputs. Compensatory cross-modal plasticity. Perhaps the most remarkable consequence of early-onset deafness is compensatory cross-modal plasticity. This is the process where brain regions normally tasked with processing the deprived sense, like hearing, are repurposed to support the processing of the remaining senses, such as vision or touch. This phenomenon is not limited to humans. It has been extensively studied in the domesticated cat. In deaf cats, behavioral enhancements related to visual processing have been documented. For example, studies involving temporary deactivation of specific auditory cortical areas in deaf cats revealed that their enhanced visual motion detection thresholds and peripheral localization abilities were mediated by the dorsal zone and the posterior auditory field, respectively. These findings provide compelling evidence that functional evolution occurs within the sensory-deprived cortex. A recent study using advanced magnetic resonance imaging, to examine regional gray matter thickness correlations in hearing and perinatally deafened cats provided fascinating structural evidence of this plasticity. By calculating the correlation of cortical thickness between 146 cortical areas, the researchers established structural connectomes for both groups. The results showed that the deaf feline brain undergoes dramatic structural reorganization. 1. Increased connectivity density. The median thickness correlation across all regions was significantly higher in the deaf cats, 0.3675, compared to the hearing controls, 0.2785. This suggests an overall increase in the density of the structurally defined connectome following deafness, with a greater number of statistically significant connections found in the deaf population. 2. Repurposing of sensory areas. This global increase in correlations was largely driven by relationships involving the auditory and visual cortices. The researchers suggest that this enhancement in interregional coactivation points toward a functional reorganization where redundancy in regional activity may develop. This structural strengthening between the auditory and visual systems aligns with the growing behavioral evidence that visual function infiltrates the deprived auditory cortex. 3. Decreased complexity. The increase in correlated activity suggests a potential reduction in the overall complexity of cortical processing. With less information to process due to the lost sense, the brain may develop these redundancies, which are likely the foundation underlying the noted behavioral enhancements in the remaining senses. 4. Connectome resilience. Despite these widespread modifications, the study found a fundamental resilience in the brain's structural organization. The majority of structural connections found in the control brain were also shared with the deaf brain. This implies that while significant compensatory adaptation occurs, the intrinsic functional organization of cortical areas is largely maintained. In conclusion, understanding deafness in dogs and cats requires navigating a complex interplay between anatomy, physiology, and genetics. Whether we are examining the high prevalence of pigment-associated deafness in Dalmatians, now linked to a specific risk haplotype near the microphthalmia-associated transcription factor gene, or the fascinating variability of dominant blue eye-associated deafness in Maine Coon cats caused by paired box3 gene variants, the message is clear. Deafness is a multifactorial challenge. Furthermore, the profound effects extend beyond the ears, forcing the central nervous system to undertake remarkable plastic adaptations, fundamentally restructuring how the brain processes the world. For clinical practice, the key remains objective diagnosis using the Brainstem auditory evoked response testing to accurately determine the extent and type of hearing loss, which, in turn, informs breeding decisions and management strategies. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.